Welcome to this course on INN and biological active substances. This online course is developed by the School of INN, which is a project within the INN program situated in the World Health Organization. This module will describe and explain the background to assigning INN to biological and biotechnological active substances. It will comprise 4 sections, an introduction followed by 3 sections covering protein and other complex biological medicines, except monoclonal antibodies, and advanced therapies. The term advanced therapies covers substances produced by sophisticated state-of-the-art technologies and includes gene therapies, cell-based gene therapies, and virus-based therapies. This section, Section A, is an introduction to INN and biological active substances. The first task is to explain what is meant by a biological medicine. Biological medicines, often referred to simply as biologicals, are medicinal products derived from living sources, such as human and animal tissue, blood, cell cultures, viruses and bacteria. Most of them are proteins in nature but include also complex carbohydrates, nucleic acids or complex combinations of them. In some cases, they may be whole microorganisms. Nowadays, most new biologicals are derived from genetically engineered cells grown in culture. These are referred to as biotechnological substances, or biotherapeutics. Compared to small molecule chemical substances, biological and biotechnological substances have a highly complex structure that may be difficult to define with absolute precision, may be heterogeneous in nature, and have many associated impurities. Biological medicines include various substances extracted from human blood such as clotting factors, immunoglobulins and hormones. Biologicals also get extracted from human and even animal tissues. Vaccines also are biologicals and may comprise a whole microorganism or virus or a subcomponent of them. Nowadays, the explosion of biotherapeutics include many enzymes for use in replacement therapies, other therapeutically useful proteins, monoclonal antibodies and totally novel bioengineered protein molecules. Biological medicines also include substances based upon viral and bacterial vectors and even comprise intact human cells. Initially used in blood transfusion and immune replacement therapy and more recently under the umbrella of cell therapy whereby various cell types including stem cells are increasingly being investigated and used in therapeutic situations. INN have been assigned to biological products since the early days of the INN program. Animal insulin preparations were first given an INN in Recommended List 3 in 1959, and in the following 2 decades in the period up to 1980, names were assigned to antibiotics, synthetic peptides, hormones and other proteins. The naming of biologicals follows the same basic rules as for chemical medicines in that, for substances related by structure and/or function, specific syllables, or groups of letters, called stems, are used to aid recognition by health practitioners and other professionals. However, names have not been assigned to natural human blood products or to traditional vaccines within the INN program. For those groups of medicines, the WHO Expert Committee on Biological Standardization, the ECBS, has assigned descriptive scientific names for these biological medicines. Recombinant proteins are those derived from cell cultures in which the cells have been genetically engineered to express a medically important therapeutic protein. These are commonly called biotechnological substances or biotherapeutics and were first approved for use as medicines in the 1980s. In 1982, the name insulin human was proposed for the recombinant protein identical to natural human insulin, and since then names have been assigned to a growing number of recombinant proteins. Since the time when insulin human became the first recommended INN, RINN, for a recombinant protein, The range of biotherapeutic substances has increased in size and complexity, and the INN program has had to develop appropriate naming schemes for new classes of medicines. The development of new classes of biotherapeutics required the creation of new naming policies and schemes. Before creating new rules, the INN program has to decide if there is benefit in providing an INN to a particular class of biological medicine. If the benefit of providing INN outweighs no INN, then an INN should be provided. If there is no obvious logic supporting a health benefit, then no INN. Thus, in the 1990s, a systematic scheme for naming monoclonal antibodies was implemented, based on the STEM map, and since then further schemes have been developed and adopted, including those for substances for gene therapies and fusion proteins. Details of these and other schemes will be addressed in separate sections of this module. New policies and schemes have to take into account the increased structural complexity of biological substances. Small molecule drugs are easily defined. Large molecular weight proteins such as antibodies can exhibit microheterogeneity such as variable glycosylation, oxidation and terminal trimming. INN can be assigned to an increasing variety of complex biological medicines including novel recombinant proteins, substances for gene therapies, with vectors based upon viruses, bacteria and plasmid DNA, virus-based therapies, nucleic acid vaccines, and substances for cell therapies, with or without genetic modification. The increase in INN requests per annum seen over the past decade or so is due mainly to requests for biologicals, with requests for biologicals now outnumbering those for chemical substances. The general policies developed for biological medicines are described in the WHO publication, INN for Biological and Biotechnological Substances, a review. Currently there are 14 general policies covering non-glycosylated substances, glycosylated substances, fusion proteins, pegylated substances, substances for gene therapies, substances for cell therapies, substances for cell-based gene therapies, virus-based therapies, monoclonal antibodies, blood products, immunoglobulins fractionated from plasma, skin substitutes, transgenic substances, vaccines. However, given that blood products, plasma-derived immunoglobulins and skin substitutes are not given INN, the policy for these is that INN are not provided for these substances. As mentioned earlier, internationally accepted names for blood products and plasma-derived immunoglobulins are agreed by a separate WHO committee, the ECBS. In addition to INN general policies for specific groups of substances, more complete naming schemes have been created for some of these groups. Thus, specific schemes for naming fusion proteins, monoclonal antibodies, and active substances for gene therapy, including cell-based gene therapy, cell therapy and virus therapy have been created. These will be described in subsections B, C and D of this module. An INN comprises a name plus a definition or description of the substance. The definitions and descriptions are equally important as the name. Chemical substances are homogeneous and can be defined by a chemical name, structure and/or formula. In contrast, to most chemical substances, biological substances have a much more complex and often heterogeneous structure, and the description of the substance is equally important as the name. Here an example of a description of a chemical substance, simvastatin, an antihyperlipidemic, is reported in the upper part of the slide. The chemical formula, the chemical name and a graphic representation of the chemical structure are provided to define the substance. In the lower part, an example of a description for a biological substance is given. The provided description of evolocumab, an antibody with hypercholesterolemic activity, describe and define the complex molecular structure of the antibody. For all proteins, further information regarding the position of disulfide bridges and of glycosylation sites are also provided in the publication of the INN. While the indication of the mechanism of action is included in the publication of the INN, it is not retained for the recommended INN and therefore it is not present in the recommended publication list. Biological substances represent the fastest growing category of medicines in the pharmaceutical world. As new classes of biological substances emerge, new naming policies will be required. Increasingly complex biological substances may require more complex names with greater use of substems and two-word names. The details of the general policies for naming proteins and other complex biomolecules, for monoclonal antibodies and for advanced therapies will be described in the following subsections. We now invite you to assess your understanding of this section answering the corresponding quiz on the School of INN website.